Hi, this is Mark Struthers. Today I'd like to talk about MR and CT acquiring data used for tissue regeneration research. We often talk about how artificial intelligence may impact on our job and what we'll be doing in the future, and I think here is a peek into the future. As imaging modalities evolve and algorithms for AI evolve, what is the future of medicine? And things are changing. One of the things we want to look at is what will the future be like in 10 years? So we're going to take a look at 3D bioprinting, which is actually regeneration of tissue. And the research today could prove to be the field of tomorrow. Medical imaging has evolved beyond the acquisition of images. Digital medical images are acquired from the interaction of energy with tissue. Ultrasound uses the energy of sound waves interacting with tissue. The rest of the modalities utilize energies that come from the electromagnetic spectrum. When we talk about imaging modalities that are currently used for bioprinting, 3D bioprinting, we utilize MR and CT to collect data. We use CT and MR because they collect data in all three planes, axial, sagittal, and coronal. And that data is interfaced into the algorithm for the bioprinting. The reason we're looking at CT and MR data is that we're looking at anatomical images only and not physiology like we'd see in a positron emission tomography scan. Digital images are created from data that can be reconstructed for a number of uses, including diagnostic interpretation, bioprinting, surgical intervention, and more. Although bioprinting can be used for many types of projects, today we're going to just focus on tissue, cellular tissue, and we need to understand the organization from cell to tissue to organ. Then the organ goes into systems, and that creates an organism or a human being. The energy that we utilize to get this data comes from the electromagnetic spectrum and is subatomical. So we have atom to molecule, molecule to cell, cell to tissue. So as you can see, we get quality data to be able to utilize for bioprinting. The acquired data is much smaller than the data for simple cell. Using MR and CT allows us to get both quality and quality of data. Currently, we can create tissues with bioprinting. The end goal is to create a functioning organ for people that might need them, such as people that need transplants. So we're going to investigate the technology today and where we would like to go in the future. Generally, groups of cells make and secrete their own support structures. These are called extracellular matrix. This matrix, or like a scaffold, does more than just support the cells. It also acts as a relay station for various signaling molecules. Thus, cell receives messages from many sources that become available from the local environment. Each signal can start a chain of responses that determine what happens to the cell. By understanding how individual cells respond to signals, interact with their environment, and organizing into tissues and organisms, we're able to create a functioning organ. To date, we've only been able to manipulate the process to mend damaged tissues or even create new ones. The process of 3D bioprinting begins with the acquisition of anatomical data, typically using imaging technologies like computed tomography and magnetic resonance imaging. These technologies generate detailed 3D images of the tissue or organ to be replicated. So as you can see, the data acquired in medical imaging is the foundation for 3D bioimaging, creating of different types of replicas and models for tissue and tissue regeneration implants. Remember, each person is unique, so we have to actually image that specific person that needs the BioPrint implant. Acquisition of medical images. The first step involves capturing detailed 2D cross-sectional anatomy images of the organ using MR or CT scans that can be made into a 3D model. Image segmentation. This step involves the use of specialized software to analyze these 2D images and isolate the organ or structure of interest from the other body features. Finally, we come to 3D reconstruction. The segmented 2D images are then processed to create a 3D model of the organ. This is done by sticking the 2D slices in the correct sequence, effectively reconstructing the organ in all three dimensions: axial, sagittal, and coronal. 3D bioprinting is often differentiated from the older scaffolding techniques for creating tissue. Scaffolding technique involves creating a temporary structure, the scaffold, that provides a supportive environment for cells to grow and differentiate. The scaffold is designed to mimic the natural extracellular matrix of the tissues, providing both structural support and biochemical cues to guide the cell behavior in the tissue generation. The materials used for the fabrication of scaffolds must possess requirements such as intrinsic biocompatibility and proper chemistry to induce molecular biorecognition from cells. 3D bioprinting is a new process that replaces the scaffolding technique. Both cells will cause tissue regeneration. Now let's take a look at 3D bioimaging and utilization of the tomographic reconstruction of the 2D images to create 3D replicas of tissue. Remember, we made a three-dimensional model of the tissue that we wanted to reconstruct to make sure the size, etc., was perfect for that particular patient. We take the 2D images that are used in the 3D reconstruction and send those to the bioprinter, which then constructs the 3D tissue layer by layer. The 3D bioprinter prints out the images layer by layer and creates a 3D model from bioinks, in other words, from living cells and tissues. The cells or stem cells are usually harvested from the patient or a compatible donor. 3D bioprinting is an additive manufacturing process that uses bioinks to print living cells developing structures layer by layer which imitate the behavior and structures of natural tissue. The integration of 3D printing into tissue engineering by utilizing live cells which are encapsulated in a specific natural or synthetic biomaterial as bioinks is paving the way toward devising many innovating solutions for key biomedical and healthcare challenges, and hopefully in the future we'll be able to create functioning organs for transplant. Now let's take a look at the 3D bioprinting process. Three-dimensional bioprinting is the utilization of 3D printing, like techniques to combine cells, growth factors, and/or biomaterials to fabricate biomedical parts, often with the aim of initiating or imitating natural tissue characteristics. Though interactive regenerative medicine is not expected to be a significant market within the next 10 years, it has and will be a major influence on the 3D bioprinting market. Bioinks are used as the base material when bioprinting tissue, organ, or bone-like structures with bioprinters. 3D bioinks can be cell-laden, scaffold-free, or cell-free, like GrowLink, which is an easily customizable hydrogel-based bioink made of nanofibrillar cellulose and water. Tissue engineering aims to develop new biofunctional tissues to regenerate and repair damaged or diseased tissue. The ultimate goal is to create scaffolds, quote unquote, that can guide the growth of new tissues and eventually degrade away, leaving behind only the newly formed tissue. The process is to load the bioink into the printer nozzle. the 3D bioprinter prints bioink layer by layer. The printed tissue is then placed in a growth media to grow and mature. Once it has matured, we have tissue ready for transplantation or other medical applications. Here we can see the real result. Here is the 3D printed bone tissue created by a scientist from Swansea University. 3D printing may provide the fabricate process to better mimic the internal and microstructures and external appearance of tissues, bones, and organs. A breakthrough in the human health industry occurred when Organovo came up with the first human tissue bioprinters. The bioprinters produced by Organovo, an American company, were capable of printing any human tissue that may be useful in regenerative therapies or medical research. This meant that it would now be possible to print out any human tissue for use in research. The bioink in the bioprinters generates human tissue using similar processes involved in additive manufacturing. The bioink is made of live human cells. By successfully mixing up the contents bioink, the bioprinter is able to generate human tissue. Using the initial results by Organovo, several researchers produced bioprinters to produce different types of human tissues. In bioprinting, we use a number of different types of bioinks and different materials in the bioinks to create the models. Take a moment to peruse this image. Take a look at the bioinks. Cross-linking, which is sometimes used after the model is formed to harden the model using either thermal energy or different types of light. We're also looking at the different types of materials used, the synthetics, the support baths, and the type of printing path that is used by the printer, layer by layer, non-planar, layer by layer, and freeform. 3D printers and 3D bioprinters are like each other, but 3D printers are designed to print solid materials, where bioprinters are designed to print a liquid or a gel. A bioprinter can be inkjet-based, laser-assisted, or extrusion-based. Each type of printer has its pros and cons when it comes to cost, cell viability, cell density, resolution, and so on. A bioprinter's compatibility with bioinks also varies, and therefore it is important to ensure that the bioprinter and the bioink work well together. 3D bioprinting can be used for several biological applications in the fields of tissue engineering, bioengineering, and material science. Clinical settings such as 3D printed skin and bone grafts, implants, and even full 3D printed organs are currently at the center of bioprinting research. The reason we have not already fabricated these tissue regeneration entities, such as the organs, is that we are not amoebas. The contents of our body are not suspended in a gooey ectoplasm. They are anchored by the extracellular matrix and, developmentally speaking, are dependent on a dizzying array of chemical cues and also physically unique to each of our bodies. Biocompatibility is key from a clinical perspective, and correct bioactivity is needed from an engineering perspective. Three-dimensional bioprinting plays an important role in tissue engineering, which aims to fabricate functional tissue for applications in regenerative medicine and drug testing. Tissue regeneration and reconstruction could enable the possibility to repair or replace damaged tissues or organs. Here we see an overview of bioprinting. We extract healthy cells from the patient. We expand the cells by culturing them in a growth medium. We then mix the cells with hydrogel or another biomaterial and create bioink. We then use the bioprinter to create new tissue. Here we see different types of tissues being used to create a structured or scaffold of a polymetric framework designed to be able to create different tissues such as the heart cartilage, and fat. Here we see a comparison between the native material and the bioprinted material for cardiac tissue, heart tissue, and adipose tissue. We take a look at the comparison, both optical and morphological. The bioprinter is more efficient in producing human tissue. Researchers are able to control the amount of tissue released during the bioprinting process. This goes a long way in saving the costs involved in the process, considering the price of bioink. The efficiency of this printer is also welcome, as it can successfully produce viable stem cells. Here we see the BioFab 4500, and also we can see some tissues that were created for surgical implants on the patient, a nose and an ear. So what are the advantages of 3D bioprinting? Well, it allows us to mimic the real structure of desired tissues and organs, etc. It has the possibility to revolutionize future medical treatment capabilities. It is possible to create a patient-specific and organ-specific treatment. You can actually test the effects of drugs, which would decrease animal testing, and we have biocompatibility with human cells and tissues. So, disadvantages of 3D bioprinting? Well, obviously, it's an expensive technology and it's also complex, and it's very difficult to maintain a cell environment. Also, there are ethical concerns. You may want to view one of my presentations on ethics in medicine, but let's just take a look at the idea that we can actually create tissues. We can go to organs, and from organs we can actually go to an organism, which would be like a human being. Throw in an AI, artificial intelligence, and now we got a robot that can work for us. That's an ethical concern. Also, the manufacturing process would take an enormous amount of energy consumption. So back to where we started. What we're looking at here is medical imaging and the use of the data collected in medical imaging and how it can be used in different fields in medicine. We just looked at one, which was 3D bioprinting and tissue regeneration. There are so many more different types of activities that we can do in the future using the data and the modalities that we have today. Here is just one company's tissue development strategy, and you can see how the technology can create lots of different products and markets. It will also be able to change medicine as we know it today. Here we see an image showing how a heart can be created for a heart transplant and saving the patient's life, and that heart would be specific to that patient. Recently, in a proof of principle study, one company created a 1 centimeter thick bioprinted tissue that was made of human bone marrow and surrounded by connective tissue and supported by an artificial endothelium-lined vasculature, which allowed the circulation of bone growth factors and subsequently the induction of bone development. As medicine evolves and digital imaging evolves along with medicine, there will be many opportunities for job growth in the future and exciting challenges for those new to the industry.